The role of a double transplant for newly diagnosed myeloma patients has been prospectively evaluated in two different clinical trials. In the EMN02 trial, which is up to date, the study enrolling the highest number of patients receiving Bortezomib-based therapy, a total of 1,500 patients were enrolled into this study that was aimed at comparing standard dose versus high dose intensification. That means VMP versus autologous stem cell transplantation for those patients in centers with a double transplant policy. A second randomization was allowed to receive a single versus a double autotransplant, and the study design also included a randomization to receive consolidation versus no consolidation therapy, VRD consolidation versus no consolidation in both arms of the study. The primary study endpoint was the progression-free survival after randomization 1, VMP versus transplant, either 1 or 2, and consolidation versus no consolidation. A secondary study endpoint was the progression-free survival from first randomization, focused only on patients randomized to a single or double transplant. This analysis was reported in New Delhi, and the results showed that patients randomized to a double transplant policy had a significantly longer progression-free survival as compared to patients who were randomized to a single autologous stem cell transplantation. This was seen in the overall patient population, although those patients who mostly benefit from double transplant were patients with poor prognosis, including patients with advanced ISS clinical stage, with high LDH, and particularly patients with a high-risk cytogenetic profile, as identified by the presence of T414 and or 1416 and or 17p deletion. In particular, in this specific subgroup of patients, double transplant was related to a two-fold longer median progression-free survival in comparison with a single autologous stem cell transplantation. I would like also to remind that another trial, the staMINA trial, which was performed in the US, gave conflicting results about the role of double autotransplant. However, the two studies are not comparable due to many differences in the design. So, if we are discussing about the results of the staMINA trial, I can recommend to take into consideration double transplant, particularly in patients who have a high-risk cytogenetic profile, as well as in patients with other poor prognosis characteristics, or in those patients who failed to achieve CR after the first autologous stem cell transplantation, although this latter issue was not addressed in the design and the objectives of the study.